welcome to plant developmental biology course. So, in previous lectures we have finished some of the example of root development. Today's class we will start with shoot development, and particularly we will focus today on shoot apical meristem maintenance. So, if you recall some of our previous lectures, so if we summarize here, so we have seen that during the development, the basic body plan which is being established during the process of embryogenesis. So, if you look at typical mature embryo, here you can see that shoot and root system has been already, the body plan, the basic body plan for root and shoot system is already established. And one very important thing what happens that the axis, body axis is established, and then the root apical meristem and shoot apical meristems are positioned along the axis. And this is, you have seen in root that this is very important for proper growth and development, particularly post embryonic development of different organs in both root system as well as in shoot system. So, in this class we will focus now on the shoot system. So, shoot system after post embryonic development, it makes a mature plant. In mature plants you have many part or many organ depending on the plant species, but if you look the basic structure, so in basic structure we have stem, some branches, some buds, then later on after transition from vegetative phase to reproductive phase, we have some structure called inflorescence, and in inflorescence we have flowers. So, these are the shoot systems part. And if you look the apex, very apex of the shoot, which is called shoot apex, it has a meristematic region, or the region where stem cells are basically positioned, stem cells which is responsible for primary growth of the shoot, and this is called shoot apical meristem. So, we will see in details what is happening and how the shoot apical meristems are maintained, but important thing here is that at the shoot apex there are coordinated processes going on. So, the coordination between stem cell maintenance as well as cell differentiation, two things are happening, and some cells, basically these cells which is in blue in color, this is basically stem cells, and this is being maintained throughout the life or till the plant is growing, and this is important. And when these cells enters in these other regions, when it leaves its the place or when it leaves its domain, it enters in the process of differentiation, and in the differentiation, depending on the identity of the meristem, it takes a different identity or different phase. For example, if the meristem identity is shoot, then it will make lateral organ for shoot, for example leaf. But if the meristem identity is inflorescence, then this will undergo the process of floral development. So, this is a typical example. You can see this is primordia, P0, P1, P2, different organs are developing, and but still a balance between cell division and cell differentiations are maintained. And this shoot basically after certain time point, it transits to the reproductive phase, and now it makes flowers. In flowers it makes some fixed organs, sepals, petals, stamens and carpel, and after making carpels in most of the flowers, this process of stem cell maintenance is inhibited, it is totally suppressed. So, that is why there is no longer maintenance of stem cells, and this is called determinate development. So, the indeterminate TC get converted into determinate development. So, this is a view of shoot apical meristem, and this is how shoot apical meristems are organized. So, this is basically inflorescence meristem. So, if you look on the top, this is how Arabidopsis, growing Arabidopsis inflorescence looks. These are the different flowers. These are early flowers, these are mature flowers where you can see that they have already come. If you look the longitudinal view, so this is how the meristem is maintained. This is the central meristem, and then you have the lateral organs coming. As I said, depending on what is the nature of meristem. If it is shoot meristem, then the lateral organs will be leaf. If it is inflorescence meristem, then lateral organs will be flowers. Both are quite similar in process of establishment and differentiation. If you look the shoot apical meristem, there are certain domains. So, these domains which you look here, this is called central zone. This is in the center of the meristem. Then you have a layer just flanking the central region, which is called peripheral zone, and then you have a domain or zone which is below the central domain, which is called rib zone. And this is the region, the green color is basically stem cell region. So, this domain is maintained, and when the cells are present in the central zone or in the stem cell niche, they will have a property of cell division. But once they leave this central domain and enters in the peripheral region or enters in the rib zone, they basically loses its stem cell property and then initiate differentiation a specific program. Another important thing, the shoot apical meristem also has different layers. So, you can have, this is the first layer which is called L1, then the second layer which is below the L1 is called L2, and then the layer beneath it is called L3 layer. So, this is the organization of shoot apical meristem, and this is again the schematic view. So, as I said that in the central zone, this region is your stem cells, and this region is also called organizing cells. This organizing cells is very, very important. So, positioning of organizing center is very important, because organizing center is equivalent to, you can say it is not exactly same, but it is equivalent to QC of root apical meristem, and it plays a very, very important role in stem cell niche positioning and maintenance. This is the scanning electron micrograph picture of the SAM. You can see this is the central region, and then these are the primordia or the lateral organs which are coming from this shoot apical meristem. If you look the establishment, when the meristem, shoot apical meristem is established, it happens at very, very early stage of embryogenesis. So, if you recall previous class, you will find that the first cell which starts cell division is zygote, and then zygote undergo the process of a series of cell division, and it undergo different stages of embryo development, and during these stages, at a particular stage, if you look here, this late stage, this region is basically assigned as a shoot apical meristem, and this region as a root apical meristem. And then you have a kind of immature seedling, if you look, shoot apical meristem is positioned here. This is picture, if you look here, this is zygote, this is single cell zygote, and this is first cell embryo. So, first cell division has occurred here, you can clearly see two nucleus. And if you look here, even the first cell division is asymmetric. So, in development, asymmetric cell division, particularly in plant development, asymmetric cell division is one very, very important process, and which is eventually giving rise to the proper developmental program or establishing proper developmental program. So, even in the zygote, you can see here the first asymmetric cell division is basically establishing the process of later development. And these processes are basically regulated by, the first asymmetric cell division is regulated by two pathways. One pathway which is mediated by YDA, which is called ADA, and another process is basically regulated by homeobox containing transcription factor WOX2, WOX8 and WOX9, and both of the pathway in the zygote, they are basically regulating the process of asymmetric cell division. Interesting thing here is that this pathway is getting activated by SSP messenger RNA, which is basically transmitted from the pollens. So, the first step of shoot apical meristem initiation, or you can say even the first state of embryogenesis, begins with this activating these two pathways, and then later on they play very, very important role. So, there is a dynamic expression and the transcript expression domain of these genes, which basically helps in in establishing apical fate of the cells or basal fate of the cells. So, if you look here, this is zygote. So, zygote basically express all three transcription factor WOX2, WOX8, WOX9. But after first asymmetric cell division, WOX2 is restricted to the apical cell, which is smaller in size, whereas WOX8 and WOX9 they retain their expression in the basal cell. Later on, you can see at this stage, at cell stage, the apical most cells they retain WOX2 expression, whereas the basal side of the apical cells they basically have WOX9 expression, and this is the reason which retains both WOX9 and WOX8, and WOX8 alone is basically maintained in this cell. So, this you can tell that the same transcription factors they are changing, or their expression patterns are basically helping in defining in establishing proper apical and basal patterning during the embryogenesis. And this is the schematic diagram you can see here. So, in basal fate after cell division at first cell embryo, the basal cell fate is basically promoted by WOX8 and WOX9, and then WOX8 and WOX9 somehow restrict WOX2 expression to the apical fate, and in WOX2 basically activate PIN1 basically, which is a kind of regulator of auxin signaling pathway. And then this is another regulators and how they basically helps in positioning the stem cell or positioning or establishing shoot apical meristem during embryogenesis. MP is transcription factor, which is auxin response factor 5. So, if you look here, the messenger RNA of MP is expressed here in the apical site, whereas messenger RNA basically it overlap here with the TMO7, whereas TMO7 protein is present here as well as here. And some other regulators of shoot apical meristem establishment and maintenance are getting also activated during the process of embryogenesis. For example, you will see later on this USHL, clavata3, CUC, STM, all these activators are basically expressed or activated at particular stage in a particular cell. Some of them are very important, which we will discuss in detail. For example, this USHL clavata signaling. USHL clavata signaling is very important, and if you look the pattern here, so if you look here, USHL is getting activated and then clavata is coming up. When clavata is coming up, USHL is restricted here, clavata is on the upper layer. So, if you look at this stage of embryo, so you can see that clavata 3 are somewhere here and USHL is pushed here. How this is happening, we will see in the next slide. And SAM establishment is basically regulated in WOX mutants. Just now I said that WOX is basically playing very important role, but if you have just single WOX2 mutant, we do not have a very strong phenotype. So, then so basically if you look the 4 mutants together, WOX1, 2, 3 and 5, then the effect is quite significant. And what you can see here, this is the expression pattern, this is in situ hybridization of clavata 3 gene. So, if you look at the very early stage of embryogenesis, you can see that it is expressed here at the tip, then it remains, it basically remains here at the upper side of the shoot apical meristem. And here you can very clearly see with the fluorescent marker how at different developmental stages of the embryo. And then if you have USHL mutant, when USHL is not present, at early stage if you see clavata 3 expression is okay, but later on the clavata 3 expression is disappearing. Okay. And if you look this mutant, in mutant basically what happens, clavata 3 is not present even from the start. So, clavata 3, which is very important regulator of shoot apical meristem, is missing in WOX mutant background. But when you put fabulosa, if you recall some of the previous class, we have little bit discussed about it. This is a class of homeobox domain containing transcription factor, and it is regulated by microRNA. So, if you put fabulosa, this is basically resistance to the microRNA. So, if you create some kind of point mutation at the site of microRNA binding, then what happens, this gene no longer will regulated by microRNA, then you can have only one regulation. And when you put back this fabulosa in VOX mutant, then what you can see that the expression of clavata3 is coming up. So, this basically tells that all these critical regulators of stem cells, they are basically playing role in shoot apical meristem. So, now if you look here, what are the key regulators which are responsible for shoot apical meristem maintenance. So, this is again just to recap you, this is the central zone, this is the rib zone, this is the peripheral zone, L1, L2 layer, and this is the lateral organs coming. And if you look some of the mutants, so this is basically wild type, you can see shoot apical meristem very nice, but if you have clavata mutant, you can see that the shoot apical meristem or the meristem is slightly enlarged. So, there is more or bigger meristem size. So, which suggests that clavata is playing some role in maybe restricting shape and maybe size of the shoot apical meristem. Wherever if you see Uschel mutant, in Uschel background you see meristem like structure, but it looks that it is terminated early. So, there is premature termination of the meristem here, whereas here is the gain of meristematic activity. But if you look this mutant, this STM shoot meristem less mutant, here you do not see any meristem. So, which suggests that STM is working at very early stage to even initiate shoot apical meristem specific program. And if you do not have STM, the meristems are not getting formed. But these genes, they are more working slightly later stage and they are more important for maintaining the meristem. So, if you look here the very general way, what happens that this clavata3 is basically expressed here, and actually the expression of clavata3 is activated by Uschel. So, if you recall, Uschel is first expressed here. So, Uschel is expressed early, then Uschel is basically activating expression of clavata3, but once it activate expression of clavata3, once clavata3 is on, clavata3 is, so basically clavata3 is a kind of signaling peptide, it is received by receptor which is clavata1, and once this signaling is activated, it inhibit expression domain of Uschel. Clavata signaling pathway ensures that Uschel expression domain or transcription of Uschel is restricted to domain of expression. And if you recall this looks quite similar to the signaling modules which we have during the root apical meristem development. There we have Cli40 is regulating the expression of VOX5, and this is quite similar. So, this is basically a negative feedback regulation. So, Uschel is activating clavata3 and clavata3 is repressing Uschel domain. Okay. Another important regulator of embryo of the shoot apical meristem is auxin, same auxin, plant hormone which is regulating almost everything. And what happens that in shoot apical meristem, the balance between auxin and cytokinin is important, and this balance is embryogenesis is regulated by VOX2 module. So, if you have VOX2 modules, the balance between auxin and cytokinin is maintained, and this maintained balance is basically required, or it is very important for regulating or coordinating or maintaining balance between cell division and cell differentiation. And if you have VOX mutants here, the balance is basically disturbed, and that is why you see the defect. Another important thing in the apical domain, so in apical domains, this is apical domain, in the peripheral of the apical domains, some of the genes are activated. One of these gene is AHP6, and AHP6 basically is the negative regulator of cytokinin signaling. And if you look here, it is very specifically expressed only in the peripheral domain of the apical regions, and in VOX mutant this expression domain is disturbed and you have AHP expression everywhere. Similarly, this is a kind of another marker which is known to express in the apical region, and in mutant background this is disappearing. So, this suggests that VOX mutants are very important in assigning a proper fate to the apical region of the meristem. So, this is, if you look the apical region, what is happening in the apical side, there are peripheral domain and the central domain. So, in central domain WOX2 module is basically getting activated, WOX2 module is regulating homeodomain G transcription factor through microRNA is also regulating, we will see maybe in the later slides. And in here in this domain, so basically WOX2 is restricting expression of AHP6 and microRNA in the peripheral domain. So, when you do not have a WOX2, that is why you can see that AHP6 expression is coming in the central domain, and this kind of pathway regulating a critical amount of auxin and cytokinin signaling pathway, and this critical balance is regulating stem cell initiation and a balance between stem cell division and cell differentiation program. Okay, so again this hormonal regulation is very important. It is extremely important for maintaining shoot apical meristem. If you look this case, so here is basically STM expression, STM expressed in the meristem, then CUC is a kind of transcription factor which regulates at the boundary of the organ. So, basically this is expressed here in the boundary between meristem and the organs. This is very important, and this boundary has to be maintained very clearly. And then you have USHL. USHL is expressed in the very center region of the meristem. If you look the responses of auxin, cytokinin and gibberellic acid, what you see that cytokinin is very dominantly present in the meristematic region, in the stem cell region, whereas the response of cytokinin in the organ primordia is very, very low. But auxin has about opposite pattern. So, if you look the meristematic region, most of the meristematic region, they do not have very high auxin signaling, whereas the primordia, the organ primordia, they have high amount of auxin signaling. Gibberellic acid is having quite similar to the auxin basis, but auxin is also having some responses here. So, this suggests that maybe cytokinin is positively regulating or very important for regulating the meristematic activity in shoot apical meristem, whereas auxin is playing major role in the process of organogenesis, starting new organ or the peripheral or the lateral organ initiation, and that is how it happens. So, if you look this is structure, this is central zone, peripheral zone. So, in the peripheral zone, if you have peripheral zone, what is happening? You have high cytokinin auxin ratio. So, you have high cytokinin and low auxin. So, that is why and gibberellic acid is low, and this pathway is basically what happening that, and STM is present everywhere. So, STM is important. STM is a positive regulator of cytokinin signaling and negative regulator of gibberellic acid signaling. So, what is happening? STM is activating cytokinin signaling in the meristematic region where gibberellic acid is low, auxin is low, then USHL is getting activated. If you have high amount of cytokinin, it activates ARR and then USHL also regulates. So, there is a kind of feedback regulation going on here. So, basically high amount of cytokinin and STM, they ensures meristematic property. But if you come in this region where the organs are coming or the site where organogenesis has to start, what is happening here? There is low cytokinin and auxin ratio. So, basically high auxin, low cytokinin. If you have high auxin, auxin is basically inhibiting STM activity there, it is inhibiting cytokinin activity, and it is probably inhibiting CUC activity, and all this process is helping in initiation of organogenesis specific program, and this is together basically ensuring that in the meristematic region or in the central region cells should be dividing, but in the region where primordia has to come, their cell division has to be restricted or the stem cell property has to be restricted, and it should be more kind of organogenesis specific property. This is another simple way of looking here. So, if you look the central region, in central region STM is activating cytokinin biosynthetic enzyme which is IPT7, and then you have high amount of cytokinin, and then cytokinin is regulating expression of cytokinin response regulators, and then it is basically helping in establishing Ushal-Clavata signaling pathway, are regulating Ushal-Clavata signaling pathway. On the other hand, if you look, ECA is auxin synthesis gene, and then you have auxin. Auxin is activating transcription factor, auxin response factor, and they are basically inhibiting here ARR activities. Loop is establishing the stem cell identity, but in the peripheral zone or in the region where you have to have a primordia, basically MP is getting activated by high auxin and STM is getting repressed, and this results in basically primordia formation in the peripheral zone. Apart from auxin and this key regulator of Ushal and Clavata signaling pathway, microRNA has been identified as a very important regulator of stem cell property or shoot apical meristem function. For example, if you have DCL1 mutant, DCL1 is basically a system which helps in biogenesis of microRNA or small RNAs. So, if you have mutant, basically microRNA pathways are getting disrupted, and if you look here in the mutant background, what happens that proper embryogenesis is not taking place, and if you look at this stage, the double mutant has unorganized growth or development. There are different types of phenotype or different classes of phenotype, but eventually what is happening that the proper organization and maintenance of shoot apical meristem is not happening in this mutant background. And if you look the auxin response or the expression of STM, because this proper auxin response has to be initiated. So, proper auxin maxima has to be achieved where it should be achieved. If that is not happening, then organogenesis will not start. At the same time, STM, which is very important regulator of meristematic activity, has to be activated for maintaining. So, both the process has to be function properly in order to have a proper balance. And if you look in this mutant background, neither proper auxin response are being generated. If you look here, auxin response is very dislocalized, or it is not at the right place. This is a kind of a typical auxin response in the wild type, but if you look in the DCL mutants, they are very diffused kind of response. It is not getting properly organized. At the same time, if you look the DCL mutant and if you look the expression of STM promoter activity in wild type, it is very nicely positioned and expressed in the right domain, but in mutant background the expression is no longer here, and this is true for another marker for shoot apical meristem. So, this suggests that when you have disrupted microRNA pathway, essentially neither stem cell is maintained nor proper organogenesis is taking place, because auxin maxima is not being generated or proper auxin responses are not getting generated here. So, this suggests that microRNA is working as one of the critical pathway to regulate the stem cell activity in shoot apical meristem. So, if we summarize here, so you can look here. So, this is a typical shoot apical meristem. You have stem cell activity going on here, then you have Ushal expression domain, Clavata 3 Ushal pathways are working here, and then Ushal is negatively regulating here ARR7, and then this is a cytokinin signaling pathway, and then LOG is basically cytokinin biosynthesis enzyme. So, STM is activating cytokinin biosynthesis through IPT7 and LOGs are getting activated here. So, eventually in this region we are having high amount of cytokinin, and this cytokinin is basically maintaining along with the Ushal and ARRs, it is maintaining here a proper stem cell property. But if you come in the lateral region or in the peripheral zone where auxin is high and auxin is basically activating transcription factor, this transcription factor MP is basically restricting the domain of cytokinin domains here in the meristematic region, but here it should be repressed so that the organogenesis process should start. And if you look here, this is slightly complex slide, but just to generally tell you that how microRNA is basically helping in establishing a proper polarity or stem cell maintenance. So, here you can see the asymmetric distribution. So, if you look this side, which is basically abaxial side, and what happens that you have high amount of microRNAs, and that is why its target, which is HDGIP, is low at this side, and if you look towards the adaxial side, it is having high amount of HDG and low amount of microRNA. So, even the gradient or the amount of microRNA and a particular transcription factor which is regulated by microRNA are very important in positioning the stem cell activity and organogenesis together and maintaining a coordination between stem cell activity as well as organogenesis. So, we will stop here. In next class, we will continue shoot development part. Thank you very much.